Hello, dear students. I welcome you all on behalf of Sri Jaindra Rank Zone classes. Dear student, we are in a continuation of the chapter that is molecular basis of inheritance. In previous lecture, we have discussed about the difference between nucleotide and nucleoside and how sugar phosphate backbone is formed. What are the different bonds are exist between them? That all we have discussed. Okay. And what are the particular components of this DNA and RNA, that is nucleic acid, and what is the history of that we have discussed in the introductory part. Today we are going to talk about the structure of DNA and then their packaging. If we talk about the structure of DNA, dear student, the full name of DNA is deoxyribonucleic acid, means it is an acidic substance. And we know that DNA is packaged in the form of chromosome within a nucleus, so it is present in nucleus. First time it was identified by Frederick Mischer in 1869. It is the first breakthrough where the identification of this nucleic acid DNA done by Frederick Mischer. He named it nuclein. He named it nuclein. However, due to technical limitation in isolating such a long polymer intact or in its original form, and the elucidation of the structure of DNA remain elusive for a very long period of time. Means the structure of DNA and its isolation was not done for a long period of time. Okay, because there was a technical limitation in the initial time period. And it is only in the 1953 the James Watson and Francis Crick, based on the X-ray diffraction data produced by Maurice Wilkin and Rosalind Franklin, proposed a very simple but famous double helical model for a structure of DNA. Which model? Double helix model for a structure of DNA. Dear student, if we talk about double helical model for a structure of DNA, okay, the initial data was X-ray diffraction data. Some images produced by this Rosalind Franklin and Maurice Wilkin, and by using the photographs of this X-ray crystallographic data, the James Watson and Francis Crick given this famous structural model of DNA. If we talk about Rosalind Franklin and a student of who is a student of Maurice Wilkin at King's College in London, they took X-ray diffraction data or X-ray diffraction pictures or images of the crystalline DNA, means solidified DNA, and they obtained very fine pictures of DNA. It was suggested that structure of DNA was a short of helix with 3.4 Armstrong periodicity, but they had not proposed a definitive model of DNA. The DNA is somewhat what helical in nature. That was obviously suggested by this Rosalind Franklin and Maurice Wilkin, but they had not proposed a definitive model of DNA. So this is somewhat X-ray diffraction pictures. But this was too much useful for the Francis Watson and Crick, and Watson and Crick in double helical model. Now, one of the hallmark of this double helical proposition or Watson and Crick proposition was base pairing between the two strands of polynucleotide chain. Means Watson and Crick proposed that there is a two strands of DNA. That two strands are polynucleotide chains, and these two strands are joined to each other, and they are joined to each other by a base pairing between the nitrogenous bases. That was proposed as one of the hallmark. However, this proposition was also based on the observation of Erwin Chargaff that for a double helical DNA, the ratio between adenine and thymine and that between the guanine and cytosine are constant and equal. So first is the X-ray crystallographic data, and second is the Chargaff's rule. Okay, if we talk about Erwin Chargaff's rule, Dear student, they also helped the Watson and Crick to identify the structure of DNA. Irvin Chargaff's role. Now, it is known that Chargaff, along with his colleagues, performed base composition studies. Performed base composition studies and put forward certain generalizations for double stranded DNA and called as Chargaff's rule. So, dear student, Chargaff not only along with his colleagues performed some of the base composition studies and put forward some of the generalization for a double stranded DNA. Dear student, you should note that this Chargaff's rule are not applicable for single stranded DNA. For single stranded DNA. And if we talk about these rules in this, purines and pyrimidines occur in equal amount. Purines and pyrimidines occur in equal amounts. Dear students, purines and pyrimidines occur in equal amount. We know that purines which are found in DNA are adenine and guanine, and pyrimidines of DNA are thymine and cytosine. So the purines, that is adenine plus guanine, is in equal number or equal amount to the pyrimidines, that is a thymine plus cytosine. And when we take a ratio of that, then Evin Chargaff showed that when we take a ratio of purines and pyrimidines, this come equal to 1. And it is known that this value is constant for all species. The next thing is, this is the ratio of purines and pyrimidine. Another ratio which we are going to talk, that is a base ratio. And when we talk about base ratio is adenine 80 base pair, means adenine plus thymine upon cytosine plus guanine. That is Cg base pair, 80 base pair upon Cg base pair, is specific for a species, is specific for each species, means is varies from species to species. So it varies from one species to another and used and used to identify these species. This ratio, if we talk, it is less than 1 in prokaryotes and, like if we take example, in E. coli it is 0.92, and it is greater than 1 in eukaryotes. It is greater than 1 in eukaryotes. If we take example, humans have 1.52. Okay, so this is the base ratio, and this the purine pyrimidine ratio is 1, but base ratio is variable from species to species. Okay, that is shown by Irvin Charkov. And in next thing, Irvin Charkov shows that sugar deoxyribose and phosphate residues occur in equal number, whereas if we talk, the purine adenine is equimolar, or means equal concentration with pyrimidine thymine. And next, the purine guanine is equimolar with pyrimidine cytosine. So these are the what Irvin Charkov's rule, and that is the generalization. Now, this two investigation helped the derivation of structure of DNA. Okay, so here we can write that X-ray crystallographic data and Chargaff's rule helped in derivation of DNA structure. Helped in derivation of DNA structure. Okay, and based on this, the James Watson and Francis Crick given a double helical model of DNA. But later on, for this structural study of this DNA, Morris Wilkin and Watson and Crick were awarded Nobel Prize in 1962, and that was shared with all three. So this is the images of the Francis Crick, the first one, then the James Watson and Morris Wilkin. The base pairing between the two strands of DNA, that is the base pairing between adenine and thymine and guanine and cytosine, confers a unique property to the polynucleotide chain. And it is known that these two genes are complementary to each other, means adenine is complementary to thymine and guanine is complementary to cytosine. So the chains are complementary to each other, and therefore, if the sequence of bases in one strand is known, if in one strand sequence of one strand is known, if suppose in sequence of one strand if A is given, then obviously we can compare that on the another strand. What will be there? T is there. If on the first strand there is a G is there, then on the another one, what we will find? C we will find. So the chains are complementary to each other, and therefore, if the sequence of bases in one strand is known, then the sequence in another strand can be predicted. Okay. Suppose if we take example, one strand is there, and they're having a DNA strands. This is a DNA, and they have a DNA have a one strand, and the DNA have an anti-parallel nature. We will study that. And they have a particularly A, T, G, C, C, G, T, A, C, and 5 dash. This is the one strand. Then on another strand, it is obviously having the opposite direction, and in place of A, the T will come. In place of T, A will come because T is complementary to A and A is complementary to T. Same way, G pairs with the C because G is complementary to C, and C pairs with the G because G and C are complementary to each other. So in place front of C, the G will come. In front of G, C will become. In front of T, A will come, and in front of A, T will come. And in front of C, G will come. It is the 3 days. So this is the one strand. If the sequence of this strand is known, then we can identify the particular sequence of another strand. Okay, so this is the particularly thing which can be observed. This is one of the, this is due to the what, a base pairing. So this provides a unique property to this. Also, if each strand of DNA, let us call it a parental strand, act as a template for the synthesis of the new strand, then the two double stranded DNA, let us call them a daughter DNA, thus produced would be identical to the parental DNA molecule. Okay, so if we have a one DNA, these two strands are going to separate and they will synthesize their copy complementary to this one. Then the two daughter DNA which will be obtained from the parental DNA will be identical to the DNA or a parental DNA molecule. So because of this, the genetic implication of the structure of DNA become very clear. Okay, and if you want to study, suppose this is the DNA having the A, T, G, C, C, G, T, A, that is 3 dash 5 dash, and suppose we are taking this one, so here it is T, A, C, G, G, C, A, T, 3 dash. So it is a DNA having a 3 dash to 5 dash and 5 dash to 3 dash. Now when replication occurs, these two strands are get separated, or copying occurs, then A, T, G, C, C, G, T, A will separate, and here this will also separate. Now on the opposite side, this new strand will be separated. So what are the complementary to this? A complementary to T. T is complementary having a G, C. So complementary to G will be C. Then here also you will find G, C, A, and T. And this is 3 dash. So here if it is a 5 dash, it is 3 dash. In front of T, what will come? A will come. In front of T, A, T will come. G, C, C, G, T, A, and 5 dash. So these are the two DNA molecule. We can call this two as a daughter DNA molecule, and this two daughter DNA which are produced that are identical to this parental DNA molecule. Okay, such a way they are produced. Now, if we talk about a structure of DNA which is based on this particular complementarity as well, that Watson and Crick proposed that DNA consists. So based on this particular all information of this X-ray diffraction data, and we can say about the Irving Chargaff's rule and some of these complementarity things, the James Watson and Francis Crick proposed that DNA consists of two strands which are helically coiled, and these two strands are helically coiled like each other. The double helix is right-handed and is referred to as B form of DNA. The double helix is coiled to the which one? Right hand side. So it is known as right-handed, and it is referred to as a B form or bDNA, and it is the most common model. DNA is formed of million of deoxyribonucleotide. Here, DNA obviously is formed of millions of deoxyribonucleotide. So, what is the structure of DNA that we need to understand? Obviously, so let's draw the structure of DNA and understand the part. So, if we draw the structure of DNA, it somewhat look like this. Here, one strand having a 5 dash, then on the another end 3 dash. Here, if it is 3 dash, on the another strand you will find a 5 dash. Okay. Why it is named as 5 dash? Because here at 5 dash and phosphate is free on the sugar. Because of this, it is known as 5 dash and means fifth carbon of ribose sugar at which phosphate is free. It is not linked to the next nucleotide. And here at third carbon of sugar, OH group is free. Now in between them, you will see like this the base pairing. They are like this. So here, if you see this particular one, it is 1 ton of DNA. And if we talk about 1 ton of DNA, in this 1 ton, it is of 34 Armstrong. Okay, it is 1 ton of 34 Armstrong, means 3.4 nanometer. And in this 1 ton of 3.4 Armstrong, 10 base pair are present. So if 10 base pair are present between these two, so if we talk about this is the base pair. And if we talk about the distance between two base pair, it is a distance between two base pair, or we can say vertical rise per base pair, then it is obviously 34 divided by 10. So it is going to be 3.4 Armstrong, means 0.34 nanometer is the distance between two base pair. Okay, and here you will see that this is known as shallow groove, and this is known as what? Deep groove. Okay, and these grooves particularly help here. In this, here you will see the base pair. That base pair can be either A double bond T or G triple bond C base pair are found. Okay, and these are the two polynucleotide chains which run anti-parallel to each other. Now, if we draw two nucleotide, suppose this part in somewhat more detail, then we will study this part in the next diagram. Okay. So if we draw that particular thing, you will see like this. The structure will look like this somewhat. So if this is the double helical model. Then if we draw this part in more detail, it will look like this. Suppose this is the ribo sugar, and here you will see OH group, and here at the fifth carbon you will see the phosphate is attached, whereas at the third carbon OH group is free, so it is 3 dash N. Now, and this is the first carbon, this is the fifth carbon, this is the first carbon. If on the first carbon, if you see the nitrogenous base thymine is attached, okay. Now this phosphate is linked to the next nucleotide, and here actually this will arrange like this. And here you will see that, and this join with phosphate. So here this is a pentose sugar, and to this pentose sugar the nitrogenous base guanine will attach, okay. And here this phosphate is free at 5 dash end, so it is, we write it to 5 dash end. Now this pair with two hydrogen bond with the thymine. Now thymine, if we talk, it is attached to the first carbon, like this. And here at the fifth carbon you will see the attachment of phosphate 5 dash N. Here the phosphate is there. Here this guanine with 3 hydrogen bond attached to cytosine. And here in cytosine you will see that sugar is like linked like this to the first carbon. And at this third, the OH group is free. Now here at this you will see that this fifth carbon, it is attached to the phosphate. Okay. So again, this is also sugar. This is also sugar. These are the nitrogenous bases. This is the 5 dash N, and this is OH, so it is 3 dash N. So this two base pair we have shown. One is TA. Sorry, here we should write A. T base pair with A and G base pair with C. So if we look, this two nucleotide will look like this. Okay, two base pair will look like this. Here the bond between the between the base two nitrogen bases will be hydrogen bond, and here you will see this is the 3 dash 5 dash phosphodiester bond. This is a phosphodiester bond. Okay. Okay. And here you will see the, that is the thymine and sugar. So we know that thymine and adenine, adenine thymine is a pyrimidine. Pyrimidine have a 1 dash 1 glycosidic linkage, whereas adenine guanine has 1 dash 9 glycosidic linkage. Here guanine has a 1 dash 9 glycosidic linkage, but cytosine have a 1 dash 1 glycosidic linkage. Okay, so this glycosidic linkage exists between this nitrogenous base and the sugar. Okay, so this way the two base pair are formed. Now further to this, okay, so this is the particularly whole structural detail of our double helical model is look like this. So this is the structure of DNA which is given by Watson and Crick. And now we are going to talk about the salient features of the double helix structure of DNA, and which are as follows. So what we seen in this double helical model of DNA, that it is made up of two polynucleotide chains where the backbone is constituted by the sugar and phosphate and bases are projected inside. Then the two chains have an anti-parallel polarity, means if one strand has a polarity 5 dash to 3 dash, then the other strand has a 3 dash to 5 dash. Then the bases in two strands are paired through hydrogen bond. Just we have seen that between the adenine and thymine, two hydrogen bonds are formed. Between the GC base pair, three hydrogen bonds are formed. So adenine forms a two hydrogen bond with the thymine from the opposite strand and vice versa. Similarly, guanine is bonded with the cytosine with three hydrogen bonds. And as a result, the purine always comes opposite to pyrimidine. And this generates approximately uniform distance because purine and pyrimidine comes always opposite to each other. So this generates approximately uniform distance, that is a 20 Armstrong between the two strands of helix. Okay, so this we have missed, that we need to note down. So let's move to the that previous slide. Okay, so yes, if we talk about the diameter between these two strands here, it is 20 Armstrong or 2 nanometer. It is going to be 2 nanometer. Okay, so that is the particularly thing happens, and that is why happening because always pyrimidine is coming against the purine and vice versa. If purine comes against the purine, then the diameter may increase compared to the 20 Armstrong, or if pyrimidine comes against the pyrimidine, then the diameter may reduce compared to the 20 Armstrong. So the particularly diameter remains constant due to this complementarity of purine against the pyrimidine. And the two chains are coiled in right-handed fashion. They are coiled right-handed. The pitch of the helix is 3.4 nanometer. A nanometer is 1 millionth of the meter, that you know 10 raised to 9 nanometer. And there are roughly 10 base pair in each turn. In each turn you will find a 10 base pair. And pitch is the what? A particularly whole turn. Consequently, the distance between the two successive base pair in helix is approximately 0.34 nanometer or 3.4 Armstrong. That is the distance between the two base pair. And the plane of one base pair stack over the other in double helix. This is addition to the plane that a one base pair stack over the another base pair in a double helix. And this plane, in addition to hydrogen bond, confers stability of the helical structure. So this particular thing help to give a whole stable structure of DNA. So now, if two purines form a base pair, being a heterocyclic, then the DNA bulged, or if the two pyrimidine forms a one base pair, being a monocyclic, the DNA become narrower. Okay, so this is the reason why purine and purine cannot be base paired, or pyrimidine cannot be base paired. So that a purine always pairs with the pyrimidine and vice versa. Therefore, the diameter of DNA remains constant. If the purine and pyrimidine start pairing, the diameter may vary. So specific purine pair with the specific pyrimidine, because of the perfect match between the hydrogen donor and acceptor site on the two bases, there is a perfect pairing. Okay. Okay, so this is all we have studied till now, that is about the structure of DNA. Now, if we talk about structure, whatever the structure we have studied, that is the form of bDNA. Now, there are different types of DNA and their comparisons. Okay, so when we talk about types of DNA, and their comparison, then dear student, there is a particularly four type of DNA, that is A, B, C, D, A. So first we are going to talk about DNA types, that is aDNA, bDNA, cDNA, and zDNA. Now, if we talk about DNA types, and on one side we take some properties, then the base pair, base pair per ton. Then we talk about a rotation, that is either they are right-handed or left-handed. Then we talk about a vertical rise per base pair and helical diameter. If we compare these four things with these properties, here if we talk about aDNA, in aDNA base pair per ton, if we talk, are 11 base pair per ton, whereas in bDNA we have studied 10 base pair per ton, whereas in cDNA they are of 9.33 and 12 base pair per ton in case of the zDNA. Whereas if we talk about the rotation is they are right-handed, then bDNA is also right-handed, cDNA is also right-handed, and zDNA is also left-handed. So this is the important question, that bDNA is a right-handed obvious, but which is the left-handed DNA? Then you should answer that it is a zDNA. Then when we count a vertical rise per base pair, then it is 2.56 base pair in case of 2.56 Armstrong actually, in case of aDNA. But in bDNA, if we talk, it is 3.4 Armstrong. We have studied that. That is vertical rise per base pair. Then it is 3.3 Armstrong in cDNA and 3.8 Armstrong in zDNA. And helical diameter, if we talk, base pair, then it is 23 Armstrong in aDNA. That is diameter between two strands. Okay, so 23 Armstrong in aDNA, 20 Armstrong in bDNA. In cDNA, it is 19 Armstrong and it is 18.4 Armstrong in zDNA. So it is a distance between two strands. Now, after this, we are going to talk about properties of DNA. Properties of DNA. Now, if we talk about the properties of DNA, dear students, we are going to discuss some of the properties, and in these properties, the first property which we are going to understand, that is a repetitive DNA. Now, when we talk about the repetitive DNA, it is a part of DNA. It is a part of DNA which contain the same sequence of nitrogenous bases which contain the same sequence of nitrogenous bases repeated more than once in genome. So if you repeat repetitive sequence or a part of DNA which has the same sequence of nitrogen base repeated more than one in a genome, then it is known as repetitive DNA. Now in these, the area with long sequence of short repetitive DNA is called satellite DNA. Now, if we want to talk about more about this repetitive DNA, then we need to talk about their types. We remember that this repetitive DNA is common in eukaryotes. Repetitive DNA is common in eukaryotes. And this satellite DNA, which we have talked, which is a long sequence of short repetitive DNA, which separate separately as in a particular ultracentrifugation. Now, if we talk about this repetitive DNA, this satellite DNA are of two types. Satellite DNA based on number of base pair involved, based on number of base pair involved. The satellite DNA are of two types. And this satellite DNA are of two types, that is microsatellite and mini satellite. If we talk about microsatellite, it is satellite DNA or a DNA with the repetitive sequences with the it has a repeats DNA repeats of 1 to 6 base pair. So these are the short repetitive sequences. But in mini satellite, if we talk, it is with more, that is 11 to 60 base pair in repeats. And this mini satellite, if we talk, used in DNA fingerprinting. Now, this repetitive sequences again are of two types. This is satellite DNA. Now, this repetitive sequences are of two types. And these repetitive sequences are of two types, that is tandem repeats and interspersed repeats. Repeats means, or tandemly repetitive sequences means, they are repeated immediately one after the other. Whereas if we talk about interspersed repeats, then here repetitive sequences are interspersed or flanked by non repetitive sequences. Means if two repetitive sequences are there and in between you will see the non-repetitive sequences, then it is interspersed repetitive sequence. Whereas if the repetitive sequence is repeated immediately, the ATGC, 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 like this, if it is getting repeated, then it is known as tandem repeats. Okay, so this is the repetitive DNA. Now, apart from this, if we talk, there is a palindromic DNA also. So another property we are going to talk, that is, after repetitive DNA, we are going to talk about palindromic DNA. Palindromic DNA. Palindromic DNA, if we talk, it is a sequence of DNA base pair. It is a sequence of DNA base pair which reads the same whether read from left to right, or right to left. If they are read in a same manner, then it is known as palindromic DNA. So it has a similar sequences on both the strand in opposite direction. So it has similar sequences in both strands of DNA. Suppose on 5 dash, if you see GAATTC 3 dash, okay, then on the another strand you will see CTT, means G complementary to C, A complementary to T, T complementary, A complementary to T, T are complementary to A, and C is complementary to G. Now if we read, then it is read like GAATTC. If we read in opposite direction, then they are read in a GAATTC. So on one strand and another strand, you will see they are particularly read in the same manner. And palindromic palindromic sequences are recognized by restriction endonuclease enzyme. So restriction endonuclease identify these palindromic sequences to cut them. Okay. Now after this we are going to talk about a third property, that is denaturation and renaturation. denaturation and renaturation. So if this is the DNA, these are the two strands of DNA, and I have hydrogen bond between these two. One is in 3 days to 5 days direction, and another one is in 5 days to 3 days direction. So it is a DNA. Now, if the breakdown of hydrogen bond occur and these two strands get separated, then it is known as a denaturation. So this is a denaturation. So what is denaturation? It is the separation of two strands of DNA from each other due to breakage of hydrogen bonds when it is exposed to high temperature, acid, or alkali. Then it is known as denaturation or melting. Now when these two separated strands again join, then it is known as renaturation. Suppose these are the two separated strands. And when these two joins, it is known as renaturation. This renaturation is also known as annealing and is annealing the reassociation of two separated strands, two separated strands by hydrogen bond formation between them. Then it is known as the particular renaturation. Now, whatever the DNA which has a more AT base pair, they can be denatured easily, and those with the GC base pair, they can be denatured slowly. So DNA with more A double bond T base pair that can be denatured more easily. So DNA with more AT base pair that is denatured more easily. So it has low melting areas, means areas which can be denatured at a low temperature easily. Whereas DNA with more GC than 80 has high melting areas and they cannot be denatured easily. Okay, so that is the what particularly denaturation and renaturation. Now after this, if we talk, the another thing which we are going to discuss here, that is the C value. C value means it is the total amount of DNA total amount of DNA per genome, and this amount of DNA this amount of DNA is expressed in picogram, and 1 picogram is equal to 10 raised to minus 12 gram. Okay, so this is the C value, that is the DNA content per genome. Per genome means whatever the total DNA content which is presenting one set of chromosome. Okay, and now we are going to talk about DNA functions. That is functions of DNA. And functions of DNA are hereditary information. Means this DNA can pass information from one generation to another generation. Then next, it is responsible for variation, and that variation arises due to the crossing over at the time of meiosis. Then C, the mutation can occur in DNA. Okay, so sudden inheritable variation due to the change in genetic material, that is due to the mutation in DNA. Then it has autocatalytic function. It has an autocatalytic function, that is DNA to DNA synthesis. Then E, it has heterocatalytic function, that is it can form DNA to RNA, proteins, hormone. All synthesis can occur from DNA. It DNA also control metabolism, growth, and differentiation. It also control metabolism specifically, and used in DNA fingerprinting. Okay, so this is the thing, that is a function of DNA which are seen. Okay, dear students, so I hope you understood this. What is this particular DNA? What is the structure of DNA? How it is made, of what it is made, what are their properties? Okay, now we remember one more important point. If suppose this is the DNA strand, which is a 3' to 5' direction, and there is the base pair. There is another, that is a 5 dash to 3 dash. This 3 dash to 5 dash strand, it is known as template strand. Then this strand, it is known as non-template strand. This non-template strand is also known as coding strand, whereas this template strand also known as non-coding strand. Now be remember the coding strand is which one, 5 dash to 3 dash. That you should remember. Okay, and template strand is in 3 dash to 5 dash direction. Other things why it is known as coding strand, that we will discuss during the transcription part, when we will study the structural part of this DNA or structure of a gene. Okay, so this is the proposition. So that is all about structure of DNA, their properties, and the different bases, and what the Watson and Crick contribution is, that all we have discussed. Okay, I hope you understand. Thank you.